Today it gives me great pleasure to introduce today's speaker, Professor Hiroaki Suga. Professor Suga goes by Hiro. So Hiro is visiting us from distant Japan, and is the first of many impressive international guests that will feature in our RNA seminar series this upcoming year. So Hiro is currently Professor of Chemistry at the University of Tokyo. He's received his bachelor's and master's from Okayama University. And he then came to the US, where he stayed for almost a decade. He first did his PhD in organic chemistry at MIT with Professor Satoru Masamune, and then moved a short distance away to be introduced to the ribosome world at the Shostak lab at Harvard Medical School. Has been combining his love and interest for organic and biological chemistry ever since. First, as an assistant and associate professor at sunny Buffalo, where he started his independent career. And then after moving back to Japan, first as professor at a research center for advanced science and technology, and then at his current department at the University of Tokyo. So Hiro is one of the most inventive and innovative modern chemists. He has successfully integrated RNA chemical biology approaches to develop new drugs. Briefly, his lab has engineered methods to modify endogenous translational systems to discover biological active macrocyclic peptides. He uses RNA enzymes that he termed flexizymes to load a natural, non-canonical amino acids to tRNA, and then he uses the ribosome itself to make, by design, macrocyclic peptides with the help of these custom tRNAs. So he has leveraged his technology to start up a company called Peptide Dreams. That was a huge success. In the way that the company was run, and the model, which has been emulated ever since by a lot of modern biotechs and startups, is that he would partner with big pharma to work on specific targets. He has stepped away from his company, and in the last 3 years, he started a new one called Mira Biologics that is even more ambitious in its scope, and I'm pretty sure it's going to be a huge success as well. So in addition to his impressive scientific accomplishment, Hiro is inspiring for multiple reasons. He finds time in his busy schedule to teach 5 courses and contribute greatly in training new scientists in Japan that he takes exceedingly seriously. So he travels extensively to give numerous talks, and he serves in multiple committees. He has received multiple awards that I don't have the time to go through right now. Instead, I would like also to highlight a little bit something that I learned from Hiro after talking to him the last few times that we met. Is he somehow finds the time to really play music with his guitar. He's really passionate about it, and Hiro has just an impressive collection of guitars in his base, including a framed autograph vinyl disc by Jimi Hendrix. So it is usually at this point that I would request you that you join me in welcoming our seminar speaker with a thunderous welcoming applause. Unfortunately, that's one of the things that we can't do anymore. So instead, what I would like to do is remind you that you can ask your questions through the Q&A. If you're feeling comfortable asking your question after the end of Hiro's presentation, then you're going to be promoted to panelists that you can ask and participate in the discussion directly. Otherwise, if you're not feeling comfortable asking your question, then I can read that for you. With that, Hiro, the virtual floor is yours, and I'm looking forward to your talk.
Thank you, Marcos. Very kind introductions for me. As introduced, I'm Professor Hiroaki Suga from University of Tokyo, the chemistry department. I know this is the University of Michigan, it's RNA community. So I will start off little different directions, but really starting from the graduate student times curiosity of the genetic code, and that's the whole start my career when I gave a thought to when I was a graduate student. So I hope that you can get sort of similar opinion or whatever you're ambitious that to go through your PhD study and get something done, what you like. So the title is Genetic Code Reprogramming that Revolutionizes the Discovery Processes of Peptides, Drug Leads. So here, the current situation of the pharmaceutical industry, what they are trying to develop. Many years, almost a century of the history developing a small molecules as a drug. These are usually you take a pill from your mouth and then it get into your body and then go circulating by the blood, and then you reach the point that your any kind of the symptom occurred by disease can be cured. The molecular weight size is about 500 Dalton. That's the kind of the range that often pharmaceutical companies could succeed developing oral available drugs. This is a sort of like empirical rule proposed by Lipinski quite some time ago. More recently, the biologics, particularly antibodies, are popular to develop the drugs. I think it's the half of the sales of the pharmaceutical drugs relied on this series of the antibodies. The molecular weight is 150,000, and generally that the antibodies are humanized, so you can avoid immunogenicity when you inject it to your body. But yet this is harder to produce sometimes, and only injectable drugs available. So lately people start calling modality and the new type of the molecules that maybe overcome the issues that the small molecule could not achieve, or sometimes the antibody could not achieve. And I have been interested in this almost 20 years of developing something different type of the molecules. And then I focused in the last 20 years developing a macrocyclic peptides, which is not really new, but it's what I want to do is I want to de novo macrocyclic peptides. The reason I want to have a macrocyclic peptide, this is a chirality rich, three-dimensional rich molecule, and then conformationally quite rich molecule. So we wanted to develop something in molecules. The molecular weight is about 1,000 to 2,500. So in the scheme of this molecular weight scale, this molecule will be, this type of the molecules are much smaller size compared to the antibodies. But yet, we wanted to have something in activities similar to antibodies, high specificity, high affinity, and high selectivity. So I just show you the view of the macromolecules. So this is an antibody binding site. This is a typical size of the protein on the left. And on the right, this is a macrocyclic peptides of which we wanted to have. And on the right, it's a small molecule. And as you see the whole entire picture, the macrocyclic peptide on the yellow is much smaller than antibodies. However, when you see the macrocyclic peptides, it's a little bit large coverage of the surface so that maybe you can achieve the similar type of activity as antibodies. We have an inspiration from the natural product. This is called cyclosporin A. Cyclosporin A was produced by fungus, and this turned out to be a very strong immunosuppressive agent for human. Of course, fungus never produced suppressive agent for humans, but accidentally this molecule turned out to be the suppressive agent. So cyclosporine A is a peptidic molecule, but if you look closely, you see the modification of the backbone with N-methyl group. So this unique modification on the background playing an important role. The cyclosporine A turned out to be the protease resistance. But moreover, there are some interesting side chain that you don't see in the protein structure, and particularly here, the D-alanine. Alanine is this configuration present in this macrocyclic peptide. And then it's macrocyclized. So all of these structural features together, cyclosporine A turn out to be very high affinity to target molecule, which is cyclophilins and a peptidase resistance. And then most importantly, membrane permeable, and this molecule was developed as oral available drugs. So it's a great molecule. We wanted to have something similar molecule like this with a variety of different targets of the proteins in order to cure the unmet medical needs. However, engineering the synthesis machinery, it's called non-ribosomal peptide synthetase, is so complex. Currently, we are unable to remodel the system and then reproduce, or something that the degenerated sequence is difficult to generate at this point. Therefore, this was, I would say, probably almost 20 years ago. I thought that I have to do something else to do. And then the system that I decided to use a translation. So I discussed today genetic code reprogramming and powered by flexizyme. Flexizyme is a flexible tRNA acylation ribozyme. I will discuss how I get it and how we developed it and how we used for the translation. And then we utilize the reconstituted translation system we call a FIT system, flexible in vitro translation system. And then finally, I will tell you about how we discover macrocyclic peptides by the platform technology we call rapid platform technology. For this audience, I won't discuss anything about translation system genetic code, as probably you know. Codon 3 bases assign a one single amino acid, and there are 20 proteogenic amino acids in the genetic code. So we have more than 20 different types of the tRNA, and the individual tRNA has an anti-codon loop. We read codons on the messenger RNA. And then 3 terminus of the tRNA hydroxyl group will be amino acylated by amino acyl tRNA synthetases specifically. So the cognate amino acids are correctly charged by ester bond formation on the 3' end of the tRNA. So my dream was to change the genetic code drastically, particularly thinking about the cyclosporine A. We need to place like N-methyl amino acid, D amino acids, and something else that we can plug in to macrocyclize. And so if you wanted to delete the genetic code and then you can replace with these new amino acids, you can simply charge amino acid on the tRNA. And the specific tRNA will read individual empty codon boxes that's shown on the left. And then you can place, let's say there are 7 codon boxes are emptied, and then you can place 7 different non-proteogenic amino acids. Particularly, we are interested in like D amino acid, N-methyl amino acid, and then even beta amino acids. Unfortunately, amino acyl tRNA synthetase are so specific to the cognate proteogenic amino acids, so we cannot use that. So we have to come up with new catalyst to do it. So that's what the flexizyme is. So I'll get into how we develop the flexizyme. Flexizyme is a very simple 45 nucleotide long of RNA molecules which catalyze the chemical reactions of the amino acid. So when it is upon a binding, let me just see. So this is the way that we developed. So initially we place random sequence at the 5' end of the transfer RNA. And then we ask a question that can we charge amino acid on the 3' hydroxyl group? So we designed this biotinylated phenylalanine cyanomethyl ester, which is a weakly activating ester group. And then we did a selection to find anything that is self-amino acid to the 3' hydroxyl group. So biotin will be tagged active sequence, so you can pull out by streptavidin agarose gel, and then you can isolate active species. And then luckily we found a single type of the sequence could catalyze the biotin phenylalaninations on the 3' hydroxyl group. So this was the start, and then we look at the sequence of this original sequence called R24, and we studied some of the unnecessary regions to truncate it down to minimize the sequence, which we call R24 mini. So at the time, we already show here the base pairing, but it wasn't very clear what the base pairing is. But we assume that maybe this 3' end is pairing with this part of the R24 mini. So to understand better of other part of the sequences, necessary sequences, we decided to maintain the ribozyme main core catalytic domain, and then randomize some part, deconnect it to the transfer RNA, and then ask the question to whether or not what kind of the molecule can be pulled out by this doped library. It turned out we are completely random sequence in here. There is no consistent sequence appeared, but here it all comes back, the original sequence. This suggested us that maybe this part is not important at all. It's rather just the 3 base pairs are critical for phenylalanine acylations on the 3' hydroxyl group. So we took X-ray structure in the collaboration of Adrian Fadmea, and we found that this 3' hydroxyl group of the tRNA is close by to the flexizyme active site. We call flexizyme. I will tell you why we call flexizyme. So we found that this is really nicely folded, and then it can charge amino acid effectively to here by this mechanism. So we wonder this simple flexizyme and can extend more diverse selectivity, means that it's actually flexible to the different type of the amino acids. I will tell you the reason why later. So we decided to re-randomize the sequence. Now we actually took the efforts, the randomizations on the amino acid binding site, which was determined by chemical mapping. And we re-randomized, then we changed the substrate. So substrate, the original substrate was cyanomethyl ester of the phenylalanine. Now we changed this benzyl group to move to the ester group. So now it's this called dinitrobenzyl ester group, but the side chain was completely non-aromatic molecule. And then here, another substrate, we made a thioester with the benzyl ester. And we tested all these substrate to reselect the peptide, and we came up and 3 different type of the flexizymes. And each flexizyme is able to specifically recognize the corresponding amino acids and then charge on the transfer RNA. But the most important part is actually the alkyl side chains are not really involved in the interaction with the flexizyme anymore. So this flexizyme turned out to be, as far as you place dinitrobenzyl ester, the amino acid can be anything. You can change the side chain to the very different type of the side chain that you never seen in proteogenic amino acids. You can place the N-methyl group, or you can put the acyl group, or you can even place the D amino acid instead of L amino acid. And then even you can place the beta amino acids. So you can put anything you want, which makes it very flexible. That's why we decided to go flexizyme. So why it's so flexizyme is interesting. So now you can change the genetic code using a flexizyme and a combination with the normal amino acid tRNA synthetases. So let's say these white letters of the amino acids are still used, but you can press flexizyme to put different type of amino acids in a genetic code. So this is just an example. You can actually design any kind of the genetic code as you wish. However, there's one technology missing, which you need to delete genetic code. You have a tool to write the genetic code, but you don't have delete the genetic code. So we use deconstituted translation system. So this custom-made translation system will be used in this way. So let's say, for example, I have only space here to discuss about 3 amino acid the reprogramming. So these 3 amino acids, we don't add intentionally to the translation system when deconstituting. And then corresponding amino acid tRNA synthetases are also removed. And we like to remove some other protein component, and like release factor 1. This is one that is important for later on in my talk. So intentionally, we make an incomplete translation system. So now you see in the view that the DNA or messenger RNA has a missing codon. The codons are there, there are the missing amino acids there. So you cannot translate peptides at all. However, if you use a flexizyme to refill these empty codon boxes with different type of the non-standard amino acids, so non-proteogenic amino acids are charged by the flexizyme. Now you actually prepared individual amino acid tRNA. The 3 tRNA will read different codon boxes, and you jump into translation system. Now you can express peptides. So that's the technology. So we come back to this rapid platform technology. So now we actually do the genetic code reprogramming, but now you need to look for drug. So how we design to do this? I just want to separate the initiation table and the elongation table for the convenience. On the left, we have 4-methionine. After that, we continue synthesis by using elongation table. So if you remove the 4-methionine or methionine, now you lose the initiation. So then we replace with a chloroacetyl D-tryptophan. This is our favorite amino acid. You can change anything you want, but this is chloroacetyl group is very important for us for the drug discovery purpose. So use a flexizyme to charge to the initiated tRNA. Now it's ready to change the initiation, so translation with this amino acid. On the right, you can actually make a library. So we make it N-U library. N means the first 4 bases, second 4 bases, a random sequence, and the last is U. So it's the red part. So all the black letters will be used for the translation. And then we remove phenylalanine, leucine, isoleucine alanine to replace with N-methylphenylalanine, N-methylserine, N-methylglycine, N-methylalanine. So we replace with the N-methylated amino acids into this genetic code. Of course, the individual amino acid could be charged to the specific tRNA. We'll read this genetic code, and that's the 4 tRNA will be ready to charge with these specific N-methyl amino acids by flexizyme, and then you are completed the genetic code reprogramming. Now you simply need the DNA synthesis. You order the synthesis with random sequence of N and T, and the length, repeat number is 10 to 15. So that means that you will translate into the peptide with 10 to 15 residues, a random sequence. So you have a variety of different lengths of the peptides. You transcribe the messenger RNA with NNU, and then we design after random sequence, we place the UGC. This is a cysteine codon. So now you remember, we start from chloroacetyl D-tryptophan, you translate according to the genetic code, and then you see some of the color amino acid would be an N-methyl amino acid, for instance. And then after the random sequence, we place the cysteine. So now you get the peptide, and N-terminus is a chloroacetyl group, and a cysteine has a side chain with a thiol, and then this spontaneously react with the chloroacetyl group to form macrocyclic thioether bond. So this thioether bond is a non-reducible bond. So it's stable in vivo. So you make a macrocyclic peptide. Turn out to be this is a quantitative yield. You get, no matter what sequence are, you always get macrocyclic molecule. So the library size of the DNA, we start from 10 to the 14th of the different sequences, go through all of these processes. At least we have 10 to the 12 different sequences. So 1 trillion different sequences became available for us. However, of course, we cannot screen 1 trillion different sequences individually. It's too many. So we use the display system. So this is actually originally, all the technique was developed by my postdoc mentor, Jack Shostak. And when you have a DNA transcribed messenger RNA and the 3' end of the sequence, we have a constant region. And then we design a DNA molecule that complementary to the 3' end of the messenger RNA. And then furthermore, DNA will be peg linked and then CC pyromycin. Pyromycin is, as you know, the antibiotics to terminate the translation. So when you mix it together, you can annihilate together. And then we design a genetic code to start from chloroacetyl D-tryptophan, and then you have random sequence with the cysteine residue. So now we're getting into a little bit updating technologies. We actually make it this genetic code reprogramming, which was not done Jack Shostak original work, but then we subjected to translation. And one more thing we invented was removing release factor 1. Release factor 1 usually comes into the termination codon and then cleaves this peptidyl bond from the tRNA by the catalysis of the ribosome. However, we remove release factor 1. Now, the ribosome does not know how to cleave this ester bond. Instead, this CC pyromycin molecule come in, and then pyromycin will be ligated effectively by the ribosome. Before this was ligation efficiency is probably about 10% or less. Now we get something like 90% of the ligation rate. So once you get this macrocyclization takes place spontaneously, now you reverse transcribe, you get the macrocyclic peptide displayed on the mRNA cDNA duplex. Now we're ready to look for macrocyclic peptide against any drug target. So we mobilize our drug target on the magnetic beads and then isolate active sequence. And once you isolate active sequence, which is extremely rare sequence, maybe a 1 to maybe 100 kinds of the active peptide can be isolated depending on the target. But our interest is after washing this, we isolate the cDNA to amplify PCR. Because we starting from 1 trillion different sequences, each round we can enrich about 1,000-fold. So we continue do the selection, and usually we run about 6 rounds of the selection. You ended up seeing a much increased the recovery rate, and then we're ready to do sequencing. And then once you know the sequence of the active peptide, we do chemical synthesis. Remember that our macrocyclic peptides are small. It's only 15 residues. So you can synthesize by chemistry very easily. Occasionally, we get only 10 residues in the macrocycle. And so it's a very facile to synthesize the macrocyclic peptides. So we can obtain large quantity. But the highlight of the profit of the rapid system and highlights in here, but the one thing I really strongly emphasize is very high cost of performance. It's only we required about 100 microliter translation for the whole entire experiment. And then you obtain active sequence information by sequencing in less than a month. So 2 to 3 weeks. My best postdoc can finish the selection under 1 week. So it's very fast. You get your target protein 1 week, you get answer. In fact, we actually have done 3 different targets of the COVID-19. We had a shutdown for a long time, and we restart our selection something like June, and then we finish July, and then we resynthesize the molecule, and then we already send them out to our collaborators now. So it's very, very quick. And then usually we get very high affinity, dissociation constant to low nanomolar to sub-nanomolar often that we can get. I just wanted to update our best technology at this point. This is quite some time ago. 2013, we reported the first time is the D-amino acid incorporation. And the D-amino acids are known that it's very difficult to incorporate. But with our technology, it turned out to be, we can see quite many of the D-amino acid could be incorporated into the peptide chain. As you see in here, what you can see that, let's say alanine case, alanine, you can migrate different length because the mobility because of the single D-amino acid changes the mobility on the page. So you can see that you can determine what the ratio of the incorporation. And there are quite many of them, as shown here, could be incorporated. So then we demonstrated the experiment with a peptide sequence, the consecutive or DLD or DLLD or DLLLD peptide synthesis. We found the last two peptide, which is the D amino acid and then 2L or 3L amino acids are inserted, then you can get the very nice expression. However, consecutive or alternate the sequence of the DL, we couldn't do the expression. So that 2013, we clearly stated in the JACs that we can't do consecutive D incorporation. Around that time, there is a very important paper appeared for the the discovery of the role of the EFP in the natural translation system by Lodina. EFP has been known for accelerating puromycin, formylmethionine puromycin ligation rate, but nothing known about what is really playing a role. And then Lodina found that the actually consecutive D proline can be significantly enhanced for the elongation by the presence of the EFP. This has stimulated us to think about maybe this is good to use for D-amino acid incorporation. So clearly the similar to proline incorporation, this was done by Kato in my group, associate professor in my group, and he actually proposed that maybe D-amino acid elongation itself is a very difficult and slow. So it's when it's slow, before the ligation occurs, this peptide tRNA drop-off has occurred. So like a proline, like the elongation. So if we can recruit EFP to this, maybe we can stop that drop-off so we can get better chance to get the amino acid elongation. So he actually even de-engineered the tRNA with a guru tRNA stem, because this actually is very good to recruit EFTU. So now this dual hybrid tRNA can recruit EFTU very well, which is the amino acid itself is actually not a great amino acid to interact with the EFTU, but this will help, and then EFP can be recruited by the D arm. So we made this tRNA, and then we expressed the peptide in the presence of EFP, which is a black, and then EFP minus is white. As you see here, we are able to elongate consecutive D amino acids much more effectively than before. And then also the presence of the EFP is really required for the consecutive D amino acid elongation. So we demonstrate two consecutive, two L amino acid, two consecutive, and so on and so forth. So we are alternating sequences. All cases, we are able to get enhancement by EFP. Let's say, for example, in the first two cases, we're unable to see any of the peptide bond formations if you don't have EFP, but in the presence of EFP, you can actually elongate it. So this changes completely our strategy, and you even can express all the peptide and the macrocyclides. So you see here is a mirror image of the D amino acids, and it can be expressed. And it's the expression level is not too far from a natural peptide expression level. So it's really a good that we can utilize this technique for the incorporation of the D amino acids. So now EFP can stimulate when tRNA pro E2, that's the tRNA, that the hybrid molecule. With this, we recently published in the Nature Chemistry in 2020, using this same approach to incorporate the cyclic beta amino acids. These again, it's extremely difficult to incorporate consecutively or even placing one amino acids into the peptide chain was hard before, but now we are able to do this. And not only we are able to do this, we actually place the 3 different beta amino acids in the genetic code, and we also reprogram the initiation with the D-tyrosine and then closed with the D-cysteine residue. So we have a 2 D amino acid and the 3 beta amino acids in the genetic code, and then we perform the selection. We construct the library that we did a selection against the 2 proteins, but I just show you today is a factor 12A, which is a very important drug target for the blood coagulations. So they're showing the outcomes. This is a peptide containing 2 beta amino acids. This is also another 2 beta amino acids incorporated. All of them are KD is a single-digit nanomolar or something like 10 nanomolar, and a Ki inhibitory activity for protease was 1 nanomolar, a few nanomolars, and completely nearly shut down the protease activity. And it's very highly selective to factor 12A. Some other factor related numbers, the protease was not inhibited, were not inhibited. So it's really specific to factor 12A. And then it's having these beta amino acids made of them is very robust against to peptidase. So it's very stable in serum. We are very happy to see the X-ray structure of the complex of the human factor 12A with the beta amino acid. This is like a folding molecule. The beta amino acid could fold peptide very well, and there are two beta amino acids. And importantly also that these amino acids, the cyclohexane ring, directly interact with the protein target. As you see here, that this is nothing really the deep pocket or anything, but it's a really nicely, a shallow site was mapped out with the macrocyclic peptide and strongly bind. So I will have a few cases that which more intensively get into drug development. So this is a Plexin B1. Plexin B1 is one of the tyrosine receptor kinases. And then it's this interactions with a semaphore D. Plexin B1 is a membrane protein, and semaphore D is a ligand. And when it binds, it's dimerize and send a signal to the, let's say, the bone cells, like osteoblast, to stop the differentiation. And so we became interested in inhibiting this protein-protein interaction and see whether you can regulate the bone homeostasis. So this is a collaboration with Professor Takagi in Osaka University. So this is the X-ray structure. We found macrocyclic peptides strongly bind to Plexin B1, KD about 3.5 nanomolar, beta sheet-like structure. It's very tightly packed, and then when it binds, and it's very tightly. We found that this macrocyclic peptide can modulate interactions with the semaphore D. Therefore, we decided to study the animal model. So in this study, we take first mouse osteoblast to see whether or not we can induce the differentiation. So this is a control mouse osteoblast cells. And then you add the semaphore D, you see the less number of the red cells. That means the differentiation was stopped. But then we add the macrocyclic peptide, we can strongly induce, even like a 50 nanomolar concentration, we really enhance the differentiation. And so we're very happy to see this result, and we decided to go to animal study. So the animal study, which in this case, we excuse from female mouse to take out ovary. In this field, the female mice that doesn't have the ovary will lose the bone strength. So we induce with we inject our macrocyclic peptides over the 13 weeks 4 times. Okay, so it's not really many injections, but over 13 weeks 4 times. And we observe whether or not we can enhance the bone strength. There are lots of quantitative data that I don't want to get into it. There are lots of examples, as you see that lots of the data was taken, but clearly you can see by bone photos. This is a wild type. And then this is the second line as the vivo, means that the female mouse bones that doesn't have the ovary. So you see the empty of the bones. And when you administrate our macrocyclic peptides, you see recovery, strongly recovered the bone strength. Even this looks like stronger than wild type. So it's a really, we are able to induce. This is actually the dose, strong enough that we can induce and to see the recovery of the strength of the bone. Okay, so next topic is a collaboration with Professor Kevin Shokat's group in the University of San Francisco, working on a K-Ras mutant of the G12D. The reason we are interested in is the K-Ras G12D is the most prevalent point mutation associated with the poor clinical outcomes for the treatment of the cancer patients. And the G12D mutation impairs both intrinsic and the GTP-ACE accelerating protein-mediated GTP hydrolysis, accumulating the GTP-bound state. This really makes a significant impact because the GTP state selective K-Ras ligand can bind to the predominant K-Ras population. Means that if we can selectively find the macrocyclic peptide inhibit this point mutant and also the GTP state selective macrocyclic peptide, we can avoid any effect for the side effect, basically. The healthy cells can express the wild type K-Ras. Unfortunately, there is no such convenient selective ligand known in the literature. So Kevin Shokat approached me to collaborate with my group to discover such a macrocyclic peptide to selectively inhibit GTP state K-Ras mutant. It's quite challenging. So we did a selection very similarly, but one thing we have to do the negative selection using K-Ras that bind from the GDP, which is actually the G12D and the T35S mutant. It turned out to be the very strongly bind to the GDP and keep the state. So we remove the macrocyclic peptide, anything that binds to this state, and then we only isolate the macrocyclic peptide that GTP-bound state. Again, if you use the GPP NHP, so GTP is uncleavable form, you can actually found one state to bind it. Although I have to say there is no clear demonstration had been made for what kind of the structure, tertiary structure appeared in each state. So we isolate macrocyclic peptide, and then we do also the repeating 5 rounds of selection. So we found 3 candidates. I showed only 2 candidates. Particularly, we are interested in this KD2, and this KD2 turned out to be selective inhibition on the GTP-bound state. So GTP-bound state interact with the LAF1, and we monitor this alpha screen methods to say the protein-protein interactions were inhibited. And the KD is about the concentration about 12.5 micromolar. Actually, KD is much tighter. It's something like a single, like 20 nanomolar range or 100 nanomolar range. But a little bit when we do this kind of the experiment, we see a little bit larger value. However, it's still inhibit, and then most importantly, this KD2, it doesn't really touch anything about of the wild type K-Ras. So it's a very selective. For instance, the Takeda peptide, which was published earlier, it did actually inhibit worse than us, but more importantly, that our peptide is much more selective. And this was much clearly revealed by looking at this experiment, which is SOS-mediated GDP exchange experiment. And our macrocyclic peptide, particularly KD1, KD2, did not really accelerate this GDP exchange. On the other hand, the Takeda peptide actually does exactly the same thing. So they are not selective for GDP form and the GTP form. So the macrocyclic peptide we isolated, it was clearly that the GTP bound state selective binder and then inhibiting interactions with LAF protein. We succeeded X-ray structure. It would turn out to be, it's very interesting macrocyclic peptide containing in the middle of water molecule to bridge the backbone. And then there's another water molecule linking together with the aspartate, which was the important the mutation occurred. One thing we are very much surprised is a structure change was made by macrocyclic peptide. When a macrocyclic peptide bound, the structure was altered, and this may be something playing an important role for the selectivity. It's not really clear yet, but we're still working on that kind of direction. And this is a more effort for the mutation. We can improve by making some bridges or unnatural amino acids that we can actually place into this. And then also we are able to see the activities up to something like below 1 micromolar. Bad news was actually this macrocyclic peptide wasn't membrane permeable. And we're still working on the membrane permeability issue. But in parallel to this experiment, we have another project, similar target. I cannot mention it right now, but it turned out to be the macrocyclic peptide was really membrane permeable. So hopefully that I can talk about that in the future. So the last topic I want to mention is the CT47 sub-alpha signaling event. This is a very important these days. Everybody know immune checkpoint inhibitor. So the cancer cells have a specific antigens, and if you identified antibodies which recognize the antigens, this antibody will be able to recruit macrophage by the interaction with the Fc gamma receptor. In this way, the macrophage can eat the cancer cells to remove the cancer cells. Unfortunately, the cancer cells are smarter. So this cancer cell has the CD47, and the macrophage has a sub-alpha. And when this interaction occurs, and the macrophage does not recognize the cancer cells as the cancer cells, and they don't eat. So this is a kind of immune checkpoint event. So we isolate the macrocyclic peptide against the SAP-alpha and then see whether we can actually destroy this SAP-alpha CD4 interactions. Now you can actually have an antibody can recruit FC gamma receptor, and the macrophage can eat cancer cells. So that's what we have done. We macrocyclic peptide. We put Alexa and then see on the surface on the cells. And these are all experiments showing here is the inhibitions between a SAP-alpha and a CD47 interactions. It's about IC50 with a 200 nanomolar range. So it's a really nice inhibitor for the SAP-alpha and the CD47. And we have already X-ray structure solved. This was just published in the Cell Chemical Biology. It turned out with the macrocyclic peptide again changed the structure of the original protein, similar to K-Ras. So these examples telling us that really macrocyclic peptide, when it binds, even changing the structure of the target protein, and then it makes a very strong contact. Again, this macrocyclic peptide containing a water molecule the inside of the macrocycle, and most importantly, when we combine together with antibodies, we are able to rescue that checkpoint event, and macrophage can start eating of the cancer cells. So we utilize 3 different antibodies: anti-CD20, anti-HER2, anti-GP75. All of them can recruit the macrophage to the cancer cells. When you didn't have our macrocyclic peptide inhibitor, you cannot get the macrophage phagocytosis. But if you add the macrocyclic peptide, we strongly enhance the phagocytosis by this way. And this is a picture that a macrocyclic peptide doesn't have any the cellular toxicity whatsoever at all. So it's really a safe molecule. Only that the event occurs in the inhibitions of the SAP-alpha and the CD47, and helping the macrophage to eat by specific antibodies. We even did for the treatment of the mouse that I don't get into details because I'm not the expert for this field, but we can actually use the antibody combinations with the macrocyclic peptide and then significantly prolong the survival rate of the mouse. And in fact, you see here that the cancer part, you see a much better shape compared to the wild type or either drugs alone. So finally, I would talk about peptidine. Peptidine was made quite some time ago, established in 2006, and IPO in 2012, and then now it's traded in a Tokyo First Stock Market, which is the largest stock market since 2015. And the market capitalization before the COVID-19 was about $7 billion. Now went down to something less than $6 billion, but still we have $5 billion worth of the company. Peptidine business model is a collaboration with the pharmaceutical companies. This is really the major our incomes for the company, and target-based contracts for macrocyclic peptide drug discovery. But there are companies that which we collaborated, and then they actually like the technology and are convinced that this technology was so worth to incorporate to their in-house programs in the company. I would say probably this is addicted by this technology, and then the peptidine will train them for the know-how of the platform technology, material transfer, and then consulting and troubleshooting, and of course, developing in-house own drugs. So in the peptide, we have a collaboration extensively with so many different companies, BMS, Amgen, and Eli Genetic, Merck in the US, and then Europe, and Novartis, and Sanofi, Janssen, Bayers, and so on. In Japan, we have quite a few companies. And then so far, 6 companies are licensed, received the license of our technology. I know very much about Merck and Genentech, what's going on, and they are really utilize our technology and to run lots of drug the screening campaign. So this is the last slide. My group, there are so many people. I'm sorry, that go back. So many people in my group who has lots of good people. It's a really a nice member of the group, and the people are come from all over the places and all over the world. And I have a great staff members, and also a great number of the postdocs and graduate students, and then collaborators, and grant agencies. And thank you very much for your attention.
Well, this is the point that we clap and applaud here for a great seminar. Thank you so much, Hiro. I guess we could use the Zoom equivalent of applaud. So this is time for questions for Hiro. So if you, I guess I'll start my video too. If you have questions, I know that Janet and Niels have questions. So you're more than welcome to ask your question, or I can read that for you. So Janet, first, if you want to ask your question.
Okay, thanks, Marcos. And thanks for a fantastic seminar. This was really amazing work and very interesting to hear about. So I have 2 questions. One, I might have missed something, and that is, how do you remove the ribozyme from after the charging reaction takes place? And the second question is, how much of these, what quantities of these cyclic peptides can you make? That is this scalable?
Okay, so the first question, we don't remove the flexizyme. So interesting, if flexizyme works with something like 100 micromolar magnesium ion concentration. Okay, so that's the most effective concentration, because it's only, we have 4 base pairs, 5 base pairs, and you need to have a high magnesium ion concentration to make it binding fine and then catalyze the chemical reactions. However, translation system is required only 10 micromolar magnesium ion. So we remove the magnesium ion. So even we actually add it together with the flexizyme in the translation system, flexizyme is not doing anything. It's invisible.
But it's not in the way?
It's not in
the way of the translation.
It's in the inside of the translation.
Yeah, but it's not a steric problem.
No,
for translation to proceed.
No, dissociate, because when you lose the magnesium ion, flexizyme is completely removed. So it's dissociated completely out and never get back.
Okay.
So we initially thought that that's an issue. So we tried to remove the flexizyme, but then later we found that this is not
okay.
at all. And the second question was how much we can create macrocyclic peptides.
Yeah.
For this is a translation we use only first round of the selection is 100 microliter, or usually we run the experiment like 5 microliter. So the amount that you produce is very, very low. It's like the concentration we reach the peptide is 1 micromolar. So you can calculate how much you produced. However, of course, you can increase the volume, you can scale up, but then its cost of the translations becomes so expensive. So we never want.
That was the reason for my question. So if you refine it down and you figure out which one is the one you want, you've got your selected one, and you want to make a lot of it, then what?
No. So when we do the selection, we have a single molecule, almost a single molecule of the one kind. Okay, but you can do the selection. You can amplify them. You can isolate them. Once you isolate them, you know the sequence. You can go to the chemistry. You can prepare chemical synthesis with a 10 kilograms, no problem.
Okay. Okay. All right.
If you want to develop the drugs, right?
Okay. So you're not using the in vitro, the reconstituted system for.
No.
Okay. All right.
No, that's not really worth to do it. Yeah.
Okay. Thanks very much.
Well, welcome.
Thank you, Janet, for your question. And Niels?
Yeah, Hiro, fantastic talk. I really was impressed. Yeah, so I had the question of whether you, in your selection, ever did in parallel, essentially, a linear peptide selection with using the same kind of technology, display, and so forth, and then basically saw how far you can get and with the linear peptide selected in the same way, and what the main differences then are. Would you, for one, get a different sequence selected? And two, I mean, what are the advantages of aiming cyclic, basically?
Yeah, so we have done in parallel in the past. We don't do that often anymore, but we have done that, and then you get almost nothing. It's criteria for the selection is definitely less than 100 nanomolar dissociation constant. And then you do linear peptide selection, it's extremely rare to get anything better than 100 nanomolar KD. On the other hand, if you do a macrocyclic peptide, you always get 10 nanomolar, 1 nanomolar, sometimes like 100 picomolar dissociation constant macrocyclic peptides. So the structure, it's a dynamic, still macrocyclic peptide is quite dynamic, but compared to the linear, is a really no comparison. And this is really the key to discover potent molecule. So unlike, let's say, RNA selection, RNA is a rather large molecule, and they're self-folded by the hydrogen bonding. So it's basically their own structure to be a macrocycle somewhat, right? You see the loops, and then the end is closed by the base pairing. So think about that, this is the way to go.
And you get this additional, I mean, so if you just use the linear peptide of the same sequence, you would lose because of entropic penalty a lot of affinity.
If you, I didn't really show anything. If you selected a macrocyclic peptide and then you linearize it, there's almost no activity. It can bind sometimes. Some cases that you see a weak binding ability, but there are lots of literature about the peptide dissociation constant micromolar. And I don't believe in almost at all. It's probably, you can measure something, but it's not really accurate numbers at all. So with our experience, if you don't get less than 100 nanomolar KD, and the peptide won't bind strong enough.
Okay, makes a lot of sense. Thanks.
Very interesting. So here, if I'm allowed just a couple of brief sort of like questions, and I know that we're like a little bit after 10 as well, so we can wrap it up after that, unless other people have questions, but they're more than welcome to hang around and ask, and if you have the time, that is. You gave us a couple of examples now of the macrocyclic peptides in crystallized with a protein, with a targeted protein, just a beautiful work. And you showed an examples of how the conformations would change of the protein conformations based at least on what was known in the absence of macrocyclic peptide. So do you know if these are conformations that are accessed natively? Are they conformations that are unnatural in the sense that they're induced by the presence of the macrocyclic peptide? Or is it that the macrocyclic peptide stabilize perhaps one that is natively accessible, but not really stable?
Oh yeah, so a case of the CD47 sub-alpha case, that where macrocyclic peptide binds, and then some part was unstructured, ripped off. And then that actually exactly happens when the CD47 binds.
Mm-hmm.
So it's basically macrocyclic peptide mimics the way that the ligand, the protein interact.
Mm-hmm.
But then it's on the other hand, the K-Ras case, according to Kevin, they never seen such a structure.
Mm-hmm.
It was quite unique when you try to find the GTP-bound form of the macrocyclic peptide, selectively binds and trap the very probably short-lived structure and effectively remove some part to make a better contact.
Mm-hmm.
So it's a case by case. I mean, you can't really define all cases, but we start to seeing this structure or these changes in the different cases now. So I think it's the early case I showed you, Plexin B1, the protein didn't change anything. It just lock and a peptide binds.
Mm-hmm.
But some cases they change the structure. Yeah.
Mm-hmm. Absolutely. So very quickly, so you've last one, sorry, and your microcyclic peptides have like a very specific length. So have you really tried to make them longer or smaller? Is there any benefit in?
Yeah, if you make it too small, we always run in parallel with a shorter peptide library. We don't mix it, but it's we run from 5 to 10, and then we run like 8 to 15 residues. So we always run both. And then usually we don't find anything excellent molecules from small library. Sometimes we do, but usually we don't. So it's clearly that some certain size is important. And if you make it longer, you basically losing diversity for exhausting diversity is lower. So eventually you don't see any significant benefit. Some parts are not used for the binding. So that's why we actually like the residues, about 15 residues at the most. So it's between 9 to 15. So we have all the mixture. So giving a chance to the macrocyclization have lots of different conformations with the different lengths and different sequences. And that's the best. And then at the end, we always want to synthesize by chemistry. Oops, it's a frozen. Anybody can answer?
I still hear you, and I can see
Okay.
you live. I don't know.
So I think Marco has issues of freeze down. So
there is actually a question in the Q&A.
Yeah, please.
Assuming that everyone else can still hear us, maybe not Marcos, but so the question here from Amitava Sinha is there any possibility that bacteria might possess flexizymes or amino acyl tRNA synthetases in some unusual form, right, RNA or otherwise, that behave as flexizymes under stress situations and come alive to help the bacterium?
Yeah, so it's really that I'm interested in putting the flexizyme in vivo in cells. Maybe then something like that could happen. I mean, I don't know whether we can do well with that. It's still a big challenge for us to get into the cell. But if we succeeded it, this is quite amazing. It's a completely new function of the new artificially selected ribozyme could function in vivo in cells.
But do you think that, there are bacteria that express certain small RNAs that actually are flexizymes naturally?
Yeah, I don't know. I don't know. We searched it for sequence similarities, and we don't get completely matched. So I'm not sure, but maybe. But it's a very interesting question.
It is an interesting question.
Yeah, thank you.
All right. Martina, I think Marcus is still not back. Yeah, so I guess it falls to me. Thanks, Hiro. This was a fantastic seminar. Let me see. I think, yeah, so that was all the questions we got so far. Fantastic seminar. I know it's late for you, late at night. I appreciate you taking the time to be with us at late an hour. And again, wonderful seminar. I think we learned a lot. It's also nice to see how the RNA world talks to the peptide world, and they are all connected, right? So it was really impressive, and how what we do in RNA science can actually find real world applications with $6 billion, $7-8 billion companies down the road, right? So anyway, so much to look forward to.
All right, thank you very much.
So thanks very much on behalf of everyone. And for the rest of the people that are still on, we have now more seminars coming up that you find in the on the web page. All right, thanks, Hiro. It was fantastic.
All right, thank you very much. Thank you. Good
Bye.
afternoon, good night.
Yep, bye.